back to our channel. This is Science Lectures 256. Don't forget to like, subscribe, and follow. And make sure you follow us on X at Science Lectures 256. So last time we looked at the bryophytes and we looked at their various characteristics. So today we are going to begin with one of the classes of the bryophytes, and that is class Hepatica. Class Hepatica, where we find our liver what's under family Muchatidae, which we shall see later. So members of class Hepatica, and hepat means liver. So these members here have a lobe-like structures analogous to that of the liver. And some of their characteristics, number they exhibit dichotomous branching, As you see here, this lobe is almost of the same size as compared to this, and whenever let's say that you add a new lobe, still a part of this will break out and it will have to grow to a certain size similar to the initial one. That's what they call dichotomous branching. Number two, they have achlorophyllous. How what they call an achlorophyllous sporophyte. So earlier we discussed and we said that the sporophyte or bryophyte is dependent on the gametophyte. So the sporophyte has no chlorophyll. That's why we call it an achlorophyllous sporophyte, and it completely depends on the gametophyte. Number three, their gametes develop directly into a sporophyte. Their gametes develop directly into a sporophyte. Their gametes develop directly into a sporophyte. Unlike as we shall be seeing later in members of class Musci, whereby they form a structure called a protonema. They form a protonema. Then their capsule is unistrous. They have a unistrous capsule. And uni means one. So their capsule is made up of one layer of cells. Then they have unicellular rhizoids. We said earlier, bryophytes do not have true roots. Why? Their root-like structures only play a role of anchorage. Yet we said typical of a root is supposed to play both anchorage and absorption of nutrients. But their root-like structures called rhizoids only play a role of anchorage, and that is why they are called rhizoids. And so these rhizoids of liverworts are made up of one layer of cells. That is why they are called unicellular rhizoids. And what we have here is the general structure of a liverwort. What you're seeing up here is our capsule, which capsule we said is unistrous or unicellular. And at the base of the capsule we have the foot. And the role of the foot is to make sure that the capsule is joined to the gametophyte. And the combination of both the capsule and the foot is what we call the sporophyte. So this is the sporophyte of a liverwort, and as you can see, it has no chlorophyll anywhere. Remember we said typical of plants they have chlorophyll, which chlorophyll is enclosed within the chloroplasts. So the capsule and the foot have no chloroplasts anywhere. That is why we say it has an achlorophyllous sporophyte, and their sporophyte is dependent on the gametophyte. Then this is our antheridium, and this is our archegonium, and the archegonium is held by what we call the archegoniopoa, and the antheridium is held by the antheridiopoa. Then these are our rhizoids, and these liverworts tend to have lens-shaped cap structures called gemma caps. So this is the outer structure of members of class Hepatica, and what we have here is just an illustration of a liverwort made up of a capsule and a foot, the antheridium, the archegonium, and these are the lobes. And these lobes analogous to the lobes of the human liver. Then these are rhizoids, and we have gemma caps. We have gemma caps. One of the characteristics is that the spores are released by elaters. Spores are released by elaters. Spores are released by elaters. And elaters, as we shall be seeing later, are hygroscopic cells that absorb water, split open to release the spores. Then, lastly, the growth of liverworts. The growth of liverworts is localized at the apex. Is localized at the apex. Localized at the apex. Why at the apex? At the apex we have the apical cell. Remember the apical cell theory, the apical meristem theory, which says that the meristem is one cell that will continue dividing but at specified points, as we shall be discussing later. So these ones have their meristems at the apical surface, and that is where growth takes place. Next, I want us to look at the reproduction in liverworts. In fact, we are going to look at the general reproduction in cryptogams. So, these cryptogams can reproduce vegetatively. They can reproduce vegetatively, and they exhibit fragmentation as a method of reproduction. Or they can reproduce sexually. They can reproduce sexually, or they can reproduce by formation of gemma. So we are going to look at everything. But generally in fragmentation, you just have the anterior portions, cells of the anterior and posterior break away from the plant and go form new plants. They go form new plants. So that is fragmentation. The cells, if this is my plant, the cells here and here shall break away, go form new plants. That is what happens in fragmentation. However, sometimes these liverworts can form gemmi, which gemmi are found in what we call the gemma caps. And these gemma caps are lens-shaped structures. They are lens-shaped structures, and these lens-shaped structures house the gemmi. And these gemmi are found when the conditions are unfavorable. That means especially during the dry season. But when wind is present, wind will force these gemma caps to open, release their gemmi, and in the presence of rain, that means we have a lot of water. These gemmi will germinate to form new crops. Will germinate to form new liverworts, and they are also common in mosses. Then, sexually, these plants exhibit what we call alteration of generations. Alteration of generations. So, alteration of generations is the existence of two distinct forms in the life cycle of an organism. Before we venture into alteration of generations, if we are trying to compare protists and fungi spend most of their time when they are haploid. Gymnosperms and angiosperms spend most of their time when they are diploid. However, for cryptogams spend some time when they are haploid, and later they will be diploid, and the cycle will continue. So we are going to see how do these plants exhibit alteration of generations. Yes, look at both the male and female structures. We say the female is made up of an enlarged portion which you call the venter, and the elongated part which you call the neck, and in the venter is where you find the egg cell. Then we say the male has a jacket of sterile cells. On top of that, it has a lead cell, which lead cell will split open, form an ostiole. Then inside here we have the androcytes, which androcytes undergo meiosis to form antherozoids. So in alteration of generations, we say that its cells will open. When they open, they form an ostiole. So this ostiole allows the antherozoids to swim free. So when this on top of that, also all the cells in the neck, that is the neck cells and the neck canal cells plus the venter canal cells disintegrate. When they disintegrate, they form what we call mucilage. And the role of this mucilage, it aids in the opening of the archegonia. So, but all this to happen, it must be in the presence of water. So what happens? We say the neck canal cells, the neck cells, and the venter canal cells and the venter cells shall disintegrate in the presence of water, form mucilage, which mucilage will force the archegonia to open. When the archegonia opens, the antherozoids will swim and enter the archegonia and fuse with the ovum, form a zygote. They form a zygote. So then this zygote shall have an extra cell wall. This zygote shall develop an extra cell wall. And when it develops an extra cell wall, we call it an oospore. It will form an oospore. Then it will be this oospore to undergo differentiation. From upper cells, it will form upper cells, and it will form lower cells. From upper cells, and it form lower cells. Then these lower cells shall undergo differentiation to form two important parts. Number one is the foot, and it will form also the seta. Then these upper cells shall form the capsule. Then this capsule, we say the role of the capsule, it encloses the spore cells. But how do we arrive at the spore cells? This capsule shall enclose a very huge mass of cells called the archesporium. So the archesporium is a conglomeration or a merger of unspecialized cells. Then these archesporial cells, shall undergo differentiation from different types of cells. Number one, they will undergo differentiation from elatas. As we have already said, elatas are hygroscopic cells that absorb water to aid in spore dispersal. Number two, these archesporial cells shall undergo differentiation from the spore mother cells. And the spore mother cells shall undergo meiosis to form tetrads. Tetrads are a group of cells a group of spores born in four. Then, at the same time, the seta shall push out of the archegonia. When it pushes out of the archegonia, it forces the archegonia to open. When the archegonia opens, it leads to the exposure of the capsule. And when the capsule is exposed, the elatas shall absorb water and aid in spore dispersal. And as we've already seen, we have the spore mother cells which will undergo meiosis to form tetrads. Two cells formed by meiosis shall form the antheridium, and two cells formed still by meiosis shall form the archegonium, and the cycle continues.